So I'll just briefly say that the rules of the game is that first I'm going to introduce I'll introduce the speakers after each one, and then we'll open it up for discussions after 10 minutes for 1 to 2. And if you won't put them into the chat, then bring them up for the discussion after all of the speakers. But our first one is Thomas Farrugia. He is at the Alaska Ocean Observing System and is the coordinator for the HAB network. He specifically has coordinated research, monitoring, and outreach. And maybe I'll just go ahead and say a few words about Dawn and Kathi, because they all fit into the harmful algal bloom. Because after, never mind. Well, let's just start with Thomas, and then when he's done, then I'll move on and introduce these speakers in order. Sorry about that. Thanks. So Thomas, do you want to start?
Yep, I'm just going through the share screening process here, and let me know how that looks. And if it doesn't look good, I can switch it, but
looks good.
Okay, great. So yeah, good morning, everybody. My name is Thomas Farrugia. I work at the Alaska Ocean Observing System, and I'm the coordinator for the Alaska Harmful Algal Bloom Network. And so what I'm going to try to do here is just kind of give you a quick background on HABs in Alaska, and then a quick word on the network itself and what it does. So algal blooms, I'm sure we're all familiar with algal blooms in general, but there are algal blooms that are beneficial and algal blooms that can be considered harmful. And so there's a definition of algal blooms, but really when we're talking about harmful algal blooms, there's kind of two main effects categories that we can think of, and one are non-toxic, things like hypoxia and mechanical impacts on fish gills, for example. But most of the time in Alaska, when we're talking about harmful algal blooms, we're talking about the toxic effects. And that's toxic effects to humans, to wildlife, and the specific effect of the toxin depends on what toxin we're talking about. So how do we get kind of from a harmful algal bloom to an impact on humans? So here's a figure from a recent paper that kind of shows all the different environmental factors that might figure into a harmful algal bloom. So, sunlight, water temperature, nutrients, anthropogenic influences, ocean acidification, and all that. Those all kind of work on the dynamics of a harmful algal bloom. But really all that, as complex as it already is only one link in the chain from HABs to humans. And that's what environmental conditions determine the presence or intensity of a harmful algal bloom. After that, we need to figure out, whether that harmful algal bloom is producing toxins and at what level, how that toxin is being taken up at the lower trophic levels, and then how that toxin is being distributed throughout the food web. And all of these dynamics might be different depending on when and where you're talking about. And Alaska being such a huge and diverse place, that can really differ from location to location. There are two main categories of kind of effects on humans that HABs might have, and I'll classify them this way. One is a food safety issue where it's a direct consumption of food items that might be toxic. And so here we're talking impacts on obviously subsistence and commercial harvests. And a lot of that is lower trophic organisms that are being consumed directly by humans, mussels, other shellfish. And a lot of those impacts have been seen in Alaska, but mostly in the Gulf of Alaska. Then there's a kind of a food security aspect to it where toxins might be affecting populations of wildlife, birds, fish, marine mammals, through either die-offs, though that's not always a clear-cut impact or a cause of the die-off, or indirect lower survivability because of the impacts on behavior, feeding, and reproduction. And all that might impact humans through food security because it threatens the food source that some communities might be relying on. So if we're going to build a program that kind of looks at harmful algal blooms, I can kind of see it as three kind of approaches or three categories that we need to do simultaneously. One, it's monitoring the algae. So what organisms are in the water? That's going to affect what toxins and what impacts we might be looking at. And this can be done with relatively low-tech approaches or really high-tech approaches. And it's really useful for understanding bloom dynamics, but it's not sufficient for determining whether or not specific food items are safe to eat. Another approach is toxin testing. So really seeing, are there toxins in certain food items or other species in the ecosystem. This does require fairly advanced techniques, usually well-equipped and labs with skilled staff. And it can aid in food safety decisions if the results are returned rapidly, obviously, to the people that have collected the samples. But it's also very helpful in HAB research to figure out how the toxins are moving through the ecosystem. And then underlying all of this, obviously, is we need good, consistent, culturally appropriate communication and outreach. Very important to increase the awareness. And eventually, if we're seeing impacts to humans, potentially this communication could hopefully help modify behaviors to limit the impacts of the toxins. So that's all the stuff we need to do, right? And so that's a lot. So we're building this slowly up through all the members that are doing all the work in the AHAB network. I'll touch real quick on the algal species. There are a lot of toxic algal species in the world. In Alaska, there's a handful, but two are really the ones that stand out most commonly, and that's Pseudonychia on the top there and Alexandrium on the bottom. Pseudonychia is a diatom. It's really thin, and it creates these chains, these long, thin chains. And it produces a toxin called domoic acid. And if ingested at high enough concentration by humans, it can produce amnesic shellfish poisoning, or ASP. I don't know of any confirmed records of ASP impacting humans in Alaska, but definitely in British Columbia and Washington State, that it is a problem that has impacted humans, and it can lead to, in the worst cases, permanent short-term memory loss. So it's very, very threatening potentially. The one that's talked about even more in Alaska is Alexandrium. It produces saxitoxins that can lead to paralytic shellfish poisoning, which can lead to death, mainly through paralyzing kind of the lungs and diaphragm and respiratory arrest. That's obviously a big problem, and we have recorded both illness and death from paralytic shellfish poisoning in Alaska. A couple of complicating factors with Alexandrium here. One is that what it produces is not just one compound, but a whole family of compound. So saxitoxins, there's over 50 congeners. That makes it more difficult to detect and to test in the lab, and almost impossible to test in the field at a kind of food safety level of accuracy. The other complication in Alexandrium, and Dr. Anderson is going to touch on this, but there's the vegetative stage of Alexandrium, which is on the left. But on the right there's a cyst stage as well. That's kind of a dormant stage that can go into the sediment and stay dormant. And so that complicates the understanding the bloom dynamics because there's kind of two stages that we need to keep an eye on and different conditions that are favorable for each one. And so those dynamics are also going to be different throughout Alaska, like I mentioned, but also the experience that there's been historically has been different. So in the Gulf of Alaska, and I'll start, oh, I'm sorry, this is going backwards. So sorry, I'll put them all up, but in the Gulf of Alaska in the south, in the green there's a relatively long history of shellfish harvest, higher population densities, and historic experience with at least PSP, if not ASP at this point. And so we have monitoring programs in place there that have been, kind of grassroots developed at the regional and local level. In the Aleutian Island and Southern Bering Sea, we have less dense human populations, still some reliance on shellfish, but also other food items. And there has been some monitoring efforts started recently, especially in places like Unalaska and the Aleutian chain. And then in the Arctic and Northern Bering Sea, historically, there's just been less experience, less contact with harmful algal blooms and their toxins, and less, if any, shellfish harvest. And really, we're talking more about impacts of the higher trophic levels. So there's a diversity there in both impacts and historic kind of experience with harmful algal blooms. This is just to give you a quick example of what can be done. This is data from the Southeast Alaska Tribal Ocean Research Consortium. So this is 17 tribes in Southeast Alaska that all have been collecting shellfish samples, blue mussels in this case, and sending them into a regional lab in Sitka to do the analyses. And you can start getting these time series of when saxitoxin concentration is the highest over time, and can give you a really good idea of when things are toxic, what conditions led to that. Some years it's in the spring, some years it's in the fall that you see the highest levels. That line there in black, horizontal line, is the FDA regulatory limit of when it would be safe to potentially consume shellfish. So you can see that there's times of the year where there's a lot of kind of heavily toxic shellfish. And so in HABs, just to finish up on this point, the concerns that there might be in the Arctic, first of all, I want to say that the Arctic Research Plan does include mention of harmful algal blooms in both priority 1 and priority 4. So that's really great and a fantastic start to start talking and bringing up capacity. But if we look at kind of how, in this case, saxitoxin might move through the food web, there's that food safety issue that might be especially important in Aleutian Islands that are still relying on shellfish that are lower trophic levels. And then in the higher trophic levels, we're talking more about wildlife impacts for the health of the ecosystem and food security of forage fish, birds, and marine mammals. I'm going to go through this very quickly because Dr. Lefebvre is, I'm sure, going to talk on it, but toxins have been found in fish, birds, and mammals throughout Alaska. So here's actually I'll just put this one up. This is actually from Dr. Lefebvre's paper. And so that's where saxitoxin domoic acid has been found in marine mammals. And then Steve Kibler at NOAA has also done similar things with forage fish or is starting to, and predatory fish. And you can see that he's found high levels of saxitoxin in those species as well. But at one point I want to make is that as far as I know, there's no evidence yet of transfer of toxins from marine mammals or fish directly to humans. And so we're not talking yet about a direct food safety issue. And so I'll finish up on this point. The AHAB network is a large network of lots of entities doing lots of very great work. And our goal is that through collaboration, we're going to address the risks of HABs to human health and wildlife populations in Alaska. We have monthly calls where we update each other. We have a website, which is getting revamped, but it's got a lot of information and resources. And we're going to build a portal to kind of show where sampling is taking place and eventually fill it out with data on phytoplankton, HAB organism locations, and toxin sampling as well. And so I'll put this up again to say thank you to all the network members. And here's my email and the URL for the website if anybody's interested. And hopefully that wasn't too long.
Oh, that's great, Thomas. Thanks so much. I'm thinking maybe after we have the two speakers who are going to go into details, we come back to the integration with the network would be great, unless somebody has a burning question right now.
I'll check the chat as well.
No, I don't see anything.
Okay.
So I think we'll come back at the end, and I think the important part on this harmful algal blooms, I mean, there's the warming seawater, we're going to learn more from Don and Kathy on that, real big impact in the Arctic, but as well as how they move through the food chain. So you kind of gave a nice introduction of where we're going on this. But Don Anderson, he's a senior scientist at Woods Hole Oceanographic. I really don't need to introduce him, but I will. He is an expert on harmful algal blooms, not just in the Arctic, but particularly in the Northeast and around the US and in other countries, specifically on toxicity, abundance, and so forth. And I think the information in his recent paper will highlight our concerns about harmful algal blooms in the Arctic. So with that, Don, you're up.
Great, thank you, Jackie. Let's see. Okay, well, Thomas has done a good job setting this up. I'm going to take you up into the sort of central and basically northern parts of Alaska. That's where the focus of our work is. Long list of co-authors here. This title, the word emerging, is a bit of a loaded term, but in fact, some of the problems that I'm going to talk about have been around for a while, for decades, in fact, maybe very episodically, maybe they're happening more frequently, but we definitely have signs that there's emerging problems, things that are getting worse, and in those cases actually likely linked to warming. Okay, so Thomas went through a lot of what HABs are, and as he said, the two syndromes, poisoning syndromes for humans that we worry about in the region I'm talking to today are the PSP and ASP problems. To make sure you get a little bit of the geography you'll hear me referring to. So I'm focusing in what we're calling roughly the Alaskan Arctic, so roughly St. Lawrence on up and into the Beaufort. Notice in the northern Bering Sea, we've got this on the western side of the Bering, we've got this Anadir Current, and then on the east side, we've got the Alaska coastal water. And sorry, my wife's phone is going off in the background. I can't do anything about it. So in the other term I want, or the other location, I want you to notice this Bering summer water, this going right through the center of Bering Strait, and then the Alaska coastal current here off towards Barrow Canyon. Notice Ledyard Bay, I'll refer to that. Point Barrow, these are a few of the locations that you'll see as we go through. Now, I won't spend time on this because Thomas did. This is the life history of Alexandrium. And as he said, much of the year, most of the year, certainly up in that Alaskan region, these cells are dormant in the sediments, and they only emerge when temperatures and other conditions are favorable to produce these vegetative cells that photosynthesize, make the toxin, and ultimately those cells form new cysts that will fall to the sediment to give you that complete cycle. So we've been very fortunate, and Jackie's been wonderful helping us and others to get a lot of data from other people's cruises, especially the DBO, the IERP, and other cruises. And that's what you'll see here, roughly covering the region that you see on the left. Fairly straightforward work with just water samples and sediment samples. So jumping to the sediment work, what this is a map of the cysts of Alexandrium catenella. That's the organism that causes the PSP problem in that region that I showed you. And in the inset there, what you see is a map of the Gulf of Maine, and it's drawn on exactly the same aerial scale and exactly the same abundance scale as what you see for Alaska. And so I've worked on this Gulf of Maine problem for ages. It's until now been considered the world's largest seed bed or cyst bed of Alexandrium, causes annually recurrent blooms and has done so for decades. And yet you look now, you can see how tiny it is. It's an order of magnitude or more smaller than what we have now characterized up in the Alaskan Arctic. So first of all, in terms of size, the feature is enormous off of the northwest coast of Alaska. Secondly, in terms of abundance, you see by the coloring here, this one cyst bed that's near Ledyard Bay and this other one up near Barrow Canyon, these are extremely dense. There's only one other place in the world with higher cyst densities, and that's actually off here to the west of the northern Bering Sea on the coast of Alaska. The numbers are also enormous. And so you've got something that's huge and something that's really dense. So to try to tell you what we think is happening with that cyst bed, here's some figures to go through. Up in the top left shows you the temperature in Ledyard Bay and near Barrow for the bottom water, and it's been binned into 5-year intervals going back then 2 decades. And what you see in general is over the last 15 or 20 years, temperatures have risen roughly 1.5 degrees. Now, if you take a look at, well, what does that mean in terms of Alexandrium? The curve on the right is generated from a lot of experimental data germinating Alexandrium cysts from a variety of areas, including the Chukchi Sea, but others as well. And all of the data falls on this beautiful red curve here, which is a degree day function. And degree days are a metric that incorporates both temperature and time. And to give you an idea of how that works really, if we incubate cysts over here, let's just say 10 degrees, it's going to take roughly 8.5 days for them to germinate. If we incubate them at 1 degree, it's going to take much, much longer. It'll take like 85 days. So you have 85 degree days in both cases. So what does that mean? We can actually take that function, these temperatures, and calculate how many more cysts are we going to produce in a bloom season. And you can see it's roughly a factor of 1.5, 1.75, almost 2. So it means we have twice as many cells, maybe twice as much toxin. It's significant, but what's potentially even more significant is shown here. These maps, the top 2 panels, show you the bottom temperatures over in the left, the decade ending in '08, on the right, the decade ending 18. So you can see how much warmer it has gotten over this whole region in the recent decade. If you subtract the one from the other, you get panel C that shows you that difference. And there you see this 2 to 4 degree warming in bottom temperature throughout the region, and you see where it's occurring. And in panel D, you see the green lines are where we have our cyst seed beds. And so we can take these temperature fields and the functions I showed you earlier and actually calculate something else, which is how long is it, well, when will these blooms actually begin in these regions? And as you see over here, it's roughly 3 weeks earlier. So 20 fewer days are needed for a cyst to germinate now than it was in the prior decade. So what does that mean? Well, these cells will take anywhere from 2 to 4, maybe 5 days to double in, basically to divide at the temperatures we've observed in surface waters in that region. That means with a 3-week head start, you can have 3, 4, 5 different generations, 5 additional generations. So the blooms, not only are we going to get more cysts coming out of the bottom, they're coming out earlier and they have much more time to grow and become abundant. All right, so looking now up to the top of the water column, top of the life history diagram, to the vegetative cells, we've gotten data that I've shown you on the right here from a number of cruises, but I'm showing you 1 cruise at the top in 2018 and 2 in 2019 that were very fortunately just a few weeks apart. So we could actually get 2 snapshots of what was going on in 2019. And what we see, and I'll go through this in more detail. If you notice in 2018, this is a bloom sitting right over that Ledyard-based cyst seed bed, and all indications were this is a locally initiating bloom from those cysts that are so abundant there. But notice in this first cruise of 2019, you see a patch of cells right north of Bering Strait. Again, very much indicative, and I could show you with all of the TS diagrams, very much associated with water that came through the strait from the northern Bering Sea. So what we've done is put together a little schematic that tries to summarize what's going on. And basically, there's 2 mechanisms for these Alexandrian blooms. The first one is one that I've said has been happening for a long time. We think actually for decades, very much episodic. It's not clear how often they have happened or how much more often they might happen as there's more flow from the Bering Sea and warmer temperatures. But in any case, these are blooms that originate someplace in the northern Bering Sea. We think it could be this western waters along the Russian coast, but it could be in the east. But one way or the other, these blooms travel through the strait, just like you saw in that 2019 cruise. The water's moving fast. Once it slows down, and that's what we see down here looking into the water, here's the bloom moving laterally with this transport system. They form cysts that fall to the bottom down here and start accumulating. But the water has been too cold historically for these cysts to germinate. And so what you have is an episodic input of cysts and a very small loss of cells through germination. And therefore you have this potential to build up and get this huge cyst abundance that we've seen. Well, what's happening now? Well, now the waters are definitely warmer at the bottom. No question, the cysts are germinating and can germinate. They can germinate, produce vegetative cells that bloom in the surface waters, make new cysts that fall out, and then you get this whole cycle in place. So you have self-initiating, so the blooms are starting there. They're self-seeding in that they're dropping cysts back again so that they have a replenishment for the next year, and annually recurrent outbreaks. That's the way it's worked in the Gulf of Maine with this exact organism, and we believe that's happening there in the northern Alaska as well. So I very quickly wanted to mention a little bit of work that's being done. This is Kate Hubbard's work with Pseudonychia up in these waters, and they basically, this huge and very complex histogram on the right shows cruises from the summer and the fall and all of the different Pseudonychia species, some of them well-known species, some of them unknowns that are still being characterized, and incredible diversity detected up there, many of them being quite toxic, or several of them being quite toxic, and a number of them being toxic. We see a mix of polar, subpolar, and temperate species. And if we look at domoic acid in the water, up in this region during some of these cruises. You can see these circles in this one summer cruise, and then over here more in the Beaufort Sea for the fall. These are low but significant concentrations of domoic acid. So to wrap up, what we've found in recent years, clearly there are multiple toxic HAB species in the Arctic, especially Alexandrium and Pseudonychus species. And these are likely to become more prevalent as warming temperatures and sea ice reduction expand that window within these blooms can initiate and persist. Now, Kathi will perhaps talk about this. We believe that the most significant risk to both ecosystems and humans is from Alexandrium catenella and the saxitoxins, in other words, the PSP problem. Pseudonychia and domoic acid are present, like I showed you, but we do consider them a second order concern at present, given the levels that we are observing of their toxins. I showed you these two extensive seed beds of Alexandrium, and bottom temperatures, no question, they're high enough to support in situ germination. I've shown you high concentrations of planktonic cells in the water column, again, with temperatures that are very supportive of that vegetative cell growth. Taken together, these data really argue that blooms in the region can originate and grow locally and are likely to be the self-seeding, self-initiating, and therefore recurrent. But also, the evidence is there that the southern populations that are transported through Bering Strait are a second source of blooms north of the strait. And so finally, this situation in the Alaskan Arctic has strong similarities with the Alexandrium distribution in the Gulf of Maine that I've studied for a long time, and where these blooms have persisted for decades. So the situation is indeed worrisome. We're on it in a sense. We are working hard, and they have some good solid support, funding support to help us. And here's a few of the funding agencies that I want to acknowledge, and lots of people who have helped with collecting samples. So I'll stop there and turn it over to Jackie and Kathi.
Sure, Dawn, thanks so much. That was really, really very interesting and concise in that. There is a question that has come up. I have my own, but I'll save that one. Is Dawn, do you think there's a causal link? This is from Thomas, a causal link between the increased temperature and the presence absence of the seed beds? In other words, is there a way for warmer temperatures at Ledyard Bay and Barrow to promote cyst deposition in those locations?
Well, the answer is yes, because those warmer temperatures, as I just showed, are clearly going to allow a faster growth rate. So you can have, in a given, all else being equal, you're going to get more cells. More cells will equate to more cysts, and therefore you would have a larger cyst seed bed. I would also say that that we have a lot of knowledge of the dynamics of Alexandrium cysts, and temperature is such a huge factor in those dynamics. And we're going through all these exercises that I showed you to estimate how many cysts and how much more the populations will be there. But it's of all the factors we look at, temperature is the one that is going to be the most significant in terms of both the cyst seed beds and the vegetative cells, because those are all coupled together.
Okay, and then a quick question: can genetics tell us more about self-seeding versus imported cells?
Wonderful, wonderful. I'm glad someone asked that question. I didn't have time to put it in there. We have several ways we are trying to look at what we'd call connectivity. And we're starting way down along the coast of Russia. We actually have a collaborative project with colleagues in Russia where they can give us samples that we can't get to. We are using two methods, one of them being microsatellites and sort of looking at molecular markers that we can hopefully start to distinguish within blooms where these cells might have come from along that whole pathway from the Bering Sea up into the Beaufort and that area. But we also have already started this. We are using toxin composition. As Thomas mentioned, there are multiple congeners of saxitoxin, and a given isolate will make maybe 5 or 6 of these, and another isolate from a different area might make 3 or 4 different ones, and it's like a fingerprint. And so we now have these cultures that we're establishing from all over the region and are starting to piece together where these cells might have come from. And I know I'm getting too long here, but the final point though, that it's going to be hard when up in the Arctic, because that's a terminal point. Everything is washing up there. So once we get up there, we're expecting to see a huge diversity of different genotypes, if you will, that are up there. Whereas as we come back south, we will see these individual populations that are more isolated. So I'm not sure whether we'll be able to resolve that as well as we might hope, but we're certainly trying.
Okay, I think there's a couple more questions, let alone my own. But if you look in the chat box, and we'll change over to Kathy's talk, but one of them, and I know the answer already, you're looking in the Northern Bering Sea too. That was one of the questions of that. What maybe when we change out, you can think about that and answer that in the chat box if you're going to look at anything in the Northern Bering Sea.
Okay.
So I think, but I think we need to go on and then come back to those questions. Okay.
All right.
Thank you. So now we're going to talk to Kathi Lefebvre, and she is a research biologist at NOAA's Northwest Fisheries Science Center and a specialist in harmful algal bloom toxins and movement and through the food web. So with that, Kathi, you're up.
Okay, great. Hi, everyone. Thank you. This is awesome. This is a great order for going through this. I'm just going to go right on from what Don was saying about the increasing risk. It's an amazing story, incredible piece of work, just really showing the risk and the growing concern. Now I'm going to tell you what we found in food webs in terms of toxin levels. So we're talking about blooms and cysts, and then we're moving into food webs. So we know that toxins accumulate in filter feeding organisms throughout the food web and have impacts on upper trophic levels. And Thomas mentioned that as well. I'm specifically going to tell you and share with you some data that we got in 2019 on cruises, same places where the data were for Don just representing. But this is all from 1 year in 2019, which was an anomalously warm year. Next slide. Can you go back one? Yes, okay, great. So this is look at this figure here. This shows a map similar to what Don was showing, but this is just a different style. We had 3 major cruises where we got samples from the food web. So Don got sediments for cysts and water samples for cell counts. We got clams, worms, fish, and zooplankton samples taken at those locations. And so this is a map sort of showing those locations. And just also, this is great maps that Rick Toman puts out showing how much above average the temperature was during that year and sampling. So you can see the sort of red waters there. Next slide. So what did we find? We looked at zooplankton samples taken from all of these locations. We looked at krill, copepods, and then a scoop of the zooplankton. So we actually had people picking out individual copepods, individual krill, and then we measured toxin levels in those organisms. And so that's what this map is showing here. I'm going to show several maps with the same exact style. So I'll take a moment to explain it here. The scale that we've shown here, the color scale, if the icon is white, it's BDL, which means below detection limit. So we did not detect saxitoxins in those samples that are white. It's just below the detection limit. Doesn't mean it wasn't there, but we didn't detect it. The yellow icons, we call those low toxin levels. And we're using the seafood safety regulatory limit to develop this scale. This is just for lack of any other way to do it. This is a metric that kind of gives us an understandable estimation of when toxin levels might be considered high or dangerous. It doesn't mean anything for the zooplankton. It only means something for human health. So just making that clear. So the yellow icons are those toxin levels that are less than half of the regulatory limit. So we're calling that low. The orange icons are those with toxin levels that are at 50% of the regulatory limit, up to the regulatory limit. And then the red, anytime you see a red icon, says, hey, that sample is at or above seafood safety regulatory limits, and for human health consumption would be considered dangerous. You would not eat that, okay? So what you can see here from the zooplankton samples is that we see low levels of toxins throughout all the areas that we sampled. We also have moderate, stay back there, sorry, moderate levels in some zooplankton samples. And then you can see there's one zooplankton sample that's red, so that's above regulatory limits. Let's flip over to the next slide. And this is showing the same type of figure with clams and worms taken from the same locations. And what you can see right away is that there is much more toxin in these benthic organisms, clams and worms, sampled from the same regions. Interestingly, anytime you had a clam or a worm sampled at the same location, the clams were always more toxic than the worms. So we're starting to understand these dynamics of which types of prey would be more potent vectors or dangerous vectors. And you'll see that there's low levels throughout in every region, in both clams and worms. There's moderate levels. And then we're at 3 regions where we have saxitoxin at or above the regulatory limit. So look at those 3 red clams off the Bering Strait, Ledyard Bay, which is right above that large cyst bed, and up in Uktiagbek. So really starting to identify that in this case, in this season, in this year, under these conditions, these are going to be more toxic vectors. This was somewhat surprising for us because remember, these clams are taken at depth. But in light of cyst bed concentrations in these areas, it starts to make some sense. So next slide. So fish, we did sample fish throughout these cruises opportunistically. These are planktivorous fish. We know that fish can accumulate toxins. Absolutely, that's for sure. But if you look at this figure from this season, this set of samples, these toxin levels were extremely low in fish. And they're even lower than they look in this figure, because I'm going to explain to you that the toxins that are shown in this figure for fish, please, are from samples taken.
I don't know what's happening. It just like automatically jumps to the next slide. So forgive me.
No worries. So these samples are from the viscera. So we took out the guts of the fish. They're not even whole fish. So these are very low levels anyway, and they're only from the viscera of the fish, not the whole fish. If you were to, and most toxin is in the viscera. Very little in comparison is in the tissues. So those are even lower if you think about eating a whole fish. That is, again, only for this time and location, likely has to do with where the fish are migrating and moving and where they are spatial and temporally with the blooms. So in other seasons, we may see something different. But in this particular situation, we adopt. Next slide. So we were also in collaboration with subsistence harvesters in Uktiagbek and on St. Lawrence Island, the villages of Gamble and Savunga. So we were able to get samples from subsistence harvested bowhead whales, you see up in the north, and we were able to get samples from walruses, which you can also see on this figure. So I just want to point out, this is just a verification, these are fecal samples, of toxin levels showing there's no question that there's exposure because we're seeing toxin levels there. Most of the bowhead whales had low levels of toxin. If you look up there, a couple of them did not have detectable levels. Walruses, on the other hand, 100% of the animals tested had at least low levels of toxin. So that's 100% prevalence. And we're seeing that in past years as well. And 2 of those animals, the orange ones, are moderate levels. And I will say that those specific concentrations in those fecal samples are almost at the regulatory limit. They're like 78 and 76 or something. So approaching what we would say, those might be high concentrations. So clearly from this sort of assessment, we know that walruses are more at risk for exposure to saxitoxin than bowhead whales, at least in this sampling year. Next. So what we can do with this information, we are getting so much data with this collaboration with all these groups working together, but we now have actual concentrations in multiple prey samples from the different regions of the Arctic. And so what I've done here in this figure is we're going to make an estimation of a daily dose of toxin that a walrus or a bowhead whale may receive eating its normal prey items, with prey items having the concentrations that we've actually measured. And what we did is we did it with average prey concentrations, and then we did it with the maximum prey concentrations, just to see what might be the maximum daily dose. And you don't need to look at all the details, but basically we use the metabolic rate of a daily requirement for a walrus or a whale. We took the caloric value of the prey items, and then we took those toxin values that we have actually measured. And for bowheads, we did krill at the average level and krill at the maximum level. And then we added the zooplankton scoop just because we had a much higher concentration there. All I want you to do now is look at that last column, daily saxitoxin dose in micrograms saxitoxin equivalents per kilogram total body weight for walruses and bowheads. And what you can see in the walrus is 5.2 and 22, but for the bowhead whales, much lower, 0.05, 0.15, 0.7. What do those doses mean? Have no idea, right? We don't know. What does that mean? What does that mean? I'm going to tell you what we tried to get from that with other information. So next slide. So this table shows different organisms and data that we have for doses of toxin that cause health impacts. The first 4 lines are for humans. These are opportunistic from actual human poisoning events. That's why the concentrations are really all over the place and very variable. But just take a look at this. Say the first line, that represents lowest observable adverse effect level from drinking water containing saxitoxin. 2.1 is the dose. Lowest dose to cause illness from shellfish consumption in one event was 21. Lethal respiratory arrest dose from shellfish you see here is in the 200 to 400. And then but a lethal dose eating crab is 1 to 2. Again, these are variable because these are not laboratory studies. These are actual what they ate at the time of that event. So that gives us an estimation. Oh, these are toxic doses to humans. If you look at mouse models and rodent models from laboratory studies, which is the next set of rows, we have a no observable effect level through gavage at 163, still no observable effect level through feeding at 958. So much higher doses are required to get effects in these mammalian rodent models. This is a well-known fact in toxicological studies. It often takes a much lower dose in larger animals compared to smaller animals. So I'm just putting that in your head so you can understand kind of how we're assessing that. There was one event that occurred with humpback whales mortality that actually Don was involved in that study. And the dose that was identified as the probable cause, it was 3.2 from eating mackerel. So now let's look at our walrus and bowhead doses. Walruses are at 22, bowheads at 0.7. Next slide. I think it's pretty clear that walruses, if not already, are certainly at risk for doses of toxin in the food web that will be harmful to the animals. Bowhead whales do not appear to be approaching that yet, at least from this 1 year of data taken from a very warm year, like obviously due to prey, metabolic demands, et cetera, locations, et cetera. So I just want to say this. Oh, next slide. That actually worked. I just want to say that this problem is not going away, and as Don has mentioned here, but this graph is showing the last 120 years sea surface temperatures in the northern Bering Sea, and the 10 warmest years are now after the year 2000. So we know that these risks are there, and we're going to be continuing to study that. And the only way to document and say that this is really happening is by getting behavioral observations of these animals at the same time, and we're working with communities to do that. And I think see, next slide. Again, the people involved are phenomenal. This is just a cast of thousands that are working with this as part of our EcoHab food web study. We do have all of this information for domoic acid as well, but Don was correct in that the concentrations of domoic acid are not at as high a level of risk as the saxitoxin are. So we focus on the saxitoxin right now. But I will say one last teaser that using ice seals as sentinels for marine mammal prevalence in the Arctic is showing that in the northern Bering Sea, there is a higher prevalence of domoic acid being present over time, not necessarily at toxic levels, but indicating that the toxins are becoming more prevalent there. So consistent with the northward expansion of pseudonychia. Thank you.
All right. Thank you so much, Kathy. There is a question, and it is, Kathy, any efforts to test birds? So I'm thinking seabirds, thinking about the benthic feeding sea ducks, the eiders.
Yes, absolutely. Yeah, and we are working very closely with the USGS, Caroline Vine Humert, other folks at Fish and Wildlife. They are sort of focusing on birds. So what we're doing is we're partnering with them, but they're sort of taking the lead on those samples and that kind of work. And then we're combining with them to share our data about what's present in the food web and how that might relate to the bird toxicity. So yes, absolutely, birds are a critical part of it.
Great. And another question, how well do we understand how long it takes for different organisms, clams, worms, fish, walruses to depurate these HAB toxins, or even if they depurate them at all? Depurate, sorry.
That's a super great question. Thank you for asking that, because I gave a really simple overview, daily toxin dose. They're getting this much in one day. Well, they're eating multiple days in a row. They're eating for weeks, and the toxins there's uptake and there's depuration. In clams and I would suspect for worms, et cetera, depuration is pretty rapid. These are water soluble toxins. So, but blooms are persistent. So if you have 3 weeks of accumulation, you're probably going to, you're going to have it, you're going to depurate it up and up and down. So that is really going to be a factor on dosing. And that's why we can't say exactly, that's why I would say being able to calculate a daily toxic dose that we just did that looks like it might have impact. And then thinking about the fact that, well, that's going to happen at least a week in a row kind of adds to the idea that they're a risk. So yes, so there is information about uptake and depuration, and it's relatively rapid. It's very rapid, and it's even in mammals.
All right, that's great.
Could I make a quick comment here, Jackie?
Yeah, I was gonna open it up just a few more. Go ahead, Don. And then I'll
just to
close
follow
it.
up on what Kathi said, though, it's notable that there are some shellfish that hang on to the toxins for a long, long time. In the Pacific Northwest, the butter clam is a perfect example where it can remain toxic a year after it was first exposed to the toxin. Others, like Kathi said, are really quick. Mussels will take it up quickly, but will also lose it quickly, and others as well. But we don't know about those depuration characteristics of some of those deeper water clams where Kathi showed that they had toxin in them. So that's something that's definitely a research area that's needed. But the worry would be that they might resemble butter clams in their long-term retention of toxins.
Right, and I would just add, because the clam part of it is that, I mean, we've kept clams alive for 5 years here in our cold room for acidification studies or looking at temperature effects and food, but it is capable to do some of those type of experiments, and they are the main food for these walruses, you know, in composite, especially the deep dwelling ones, the 50-year-old ones. You know, there's the Seripes and Mayas that are going down deep, that are suspension feeding, are all capable to just build these toxins up if they're able to stay. It seems to me. My question to you, and I'm going to check anymore, is how many times can those cysts, you know, they go down, they come back up. Can they do multiple life cycles over a couple of months?
Well, up there, I don't think so. But in fact, we in the Massachusetts area, believe it or not, we were just finding Alexandrium cells in the water of one area we studied in January. It's sort of like, it's supposed to be ice on the water, even on the salt ponds and so forth, but it's been a fairly warm winter. But so what we're seeing in some parts of the world are 2 pulses during the year, one in the spring, early summer, and then there's another one now that we're seeing in the fall and early winter. And as it's warming in our region, I think that second one can start to become more important. I think things are still so cold up there that that's not likely yet. I mean, it might be a long time before that, but what I think we're seeing is that first pulse is really starting to become significant given the temperatures.
Okay, great. I mean, I think there are more questions. I'll talk to you some other time. So, because I think we need to close up if we want to keep this, because I do want to give Carin Ashjian to get some community updates and some activities before but I do want to thank all the speakers, to Dawn and Kathi and Thomas for a great group of presentations. And there's more things coming in, questions, so take a look at the chat. So at this point, I think I'd like to have Carin Ashjian to be able to say a few words about the Synoptic Arctic Survey. And Carin is presenting on, Carin is a senior scientist at Woods Hole Oceanographic Institution and a specialist biological oceanographer on zooplankton and ecosystem dynamics. So do you want to share there, Carin?
Sure.
Thanks,
Jackie. Thank you, everybody. I'm just going to give you a brief update, as Jackie mentioned. Oh, gosh, I hope this works. I just realized I've got this big screen on here.
Yeah, we see it. You just put it in show mode.
Yeah, I'm putting it into, but it takes a second. I forgot to unplug the ancillary screen, so it's very slow, but hopefully it will come up. Do you see it?
We see it. You see your notes.
Okay, hang on. Sorry about that. I have a big screen and it doesn't play nicely with
the other one.
with PowerPoint, you know. Okay, now do you still see it?
Yes, that's perfect. Go ahead.
Yeah, okay, I unplugged the other screen. Okay, so yeah, I'm just going to give a brief update on the Synoptic Arctic Survey, on the US component of the Synoptic Arctic Survey. The Synoptic Arctic Survey is an international program. And Jackie and I are the leads for the United States effort. And we are also the lead PIs on this project and the co-chief scientists for a cruise that has been funded by the US National Science Foundation. This has been a long time coming, but we've just heard within the last couple of weeks that our cruise has been scheduled on the Coast Guard cutter Healy for this year. So it's a bit of a scramble. The cruise starts in late August. It's a 50-day cruise, and the plan is to go to and from Dutch Harbor. And on the left is the draft cruise track. And I notice I say draft, and then I have not final all over the place. And also caveats about planning discussions have just started because we really have only started talking with the Coast Guard about the cruise. We had one meeting with them so far. But the lines on the left show the draft cruise track that we had put in our proposal. And the logic behind the two tracks is that the eastern track goes across the geotraces track, and the western track, which was at 2015, the western track goes across the Trans-Arctic section track, which took place in 1994. And of course, geotraces did not sample the same things except for some of the physical parameters that the Synoptic Arctic Survey is designed to sample. But of course, the physics will be similar, and the Trans-Arctic section did sample some of the biological components that are very rarely sampled in this region. The work for this project is supposed to start in the northern Chukchi Sea, so that bottom. This is this was actually written for a non-specialist audience, but it describes the different things that we have included in our project to sample. You can see it ranges from some of the physical characteristics through the nutrients, chlorophyll, zooplankton distributions, and some rate processes, benthic work, a whole bunch of different carbon, because the carbon system is one of the targets of the Synoptic Arctic Survey effort. And then we are going to also hope to include marine mammal distributions and seabird distributions. An important note for the broader scientific community is that we will have all underway sensors on the Healy running and collecting data during this effort. So even there will be opportunities for people to later write proposals to use those underway sensor data. The geophysics is really important in this region. I don't think there's a whole lot of work done up there. So there's lots of going to be opportunities later for work with the data from those other sensors. And this is my last slide. I'm just going to say more information is going to be coming. As I mentioned before, we're working with the Coast Guard on the cruise planning, finalizing the cruise dates and cruise track. We don't know if they'd have any extra berths for which we could recruit additional Synoptic Arctic Survey projects. But we will hold a town hall at the upcoming Ocean Sciences meeting, and we hope to have more information to present at that time. And this is a cute picture from 2014, and pulling the Healy would be very slow indeed, but we'll get there eventually. So thank you for your attention, and I'm happy to answer any questions.
Thank you, Carin, so much. We will stay on 5 more minutes. If anybody has any questions for Carin on here, and otherwise I'm going to pass it to Danielle in a few minutes to close the meeting up and answer any other further questions. Here, does anybody have any questions for Carin? Let's see. No, nope. I think these are all HAB ones. Okay, so I think the town hall that's going to come in the Ocean Sciences meeting, it's an online meeting. Unfortunately, I think you have to be registered to be listened to it, but the Arctic Science Summit Week at the end of March will be free for any online participants, and we will have a longer workshop there on the SAS, just to give you a heads up. So at this point, I want to appreciate all the speakers that we had, and once again, thanks so much. I think there's some interesting things coming in the chat and connections, hopefully. And Danielle, I'll let you close this webinar up.
Thanks so much. This really was a wonderful event. Thank you so much to everyone for participating, and thank you to our speakers. Apologies for the cuckoo clock in the background. I wanted to mention that our Marine Ecosystems Collaboration Team has decided to shift the timing of our monthly meeting as we move into the next 5-year Arctic Research Plan. We'll now be meeting at the same time Wednesdays at 10 a.m., but the second Wednesday of every month instead of the 4th. So please mark your calendars. Please come participate with us on a monthly basis. We would love to have you join us the second Wednesday of every month at 10 a.m. Alaska time. And what we are planning right now to host another webinar next month in March to seek input from our research community about how IARPC will implement the new 5-year research plan. So they're working on an implementation plan that's going to include some specific objectives and deliverables. And we'd love to hear from our team members your ideas about how you would like to see our team contribute to implementing the new Arctic research plan. So I encourage you to join us next month and think about that between now and then. And we look forward to seeing you next month. Thank you so much to everyone for participating.
Yes, thank you. All right, I think didn't have to extend too much longer than the hour. So appreciate you.
Thank you.
Bye-bye.
Bye.
Thank you.